Hi, my name is Noah Hopkins. I'm presenting on behalf of myself, Nina Parvari, and Sania Aziz, and we are presenting over the WFS1 gene variants that affect sensory neural hearing loss. In our literature-based project, we're seeking to know why and how mutations of the WFS1 gene can cause sensory neural hearing loss. Sensory neural hearing loss is caused by damage to sensory nerves, and it can be caused by many different things. Um, in particular, the WFS1 gene is a gene that causes Wolfram syndrome, affecting many systems, including hearing. One way that it affects hearing is by affecting the calcium gradient regulation in the cochlea. WFS1 encodes wolframin, which is a protein that regulates the endoplasmic reticulum stress response and calcium uptake at the membranes. Calcium ions are really important for auditory signal transduction. Existing literature shows that mutations at this gene can alter wolframin's shape, which can affect the membrane receptors and cause calcium uptake reduction in cochlear cells. The resulting disruption can impair signal transduction, which leads to hearing loss. What is not yet known is how many mutations exist in this gene. A 2020 study presented a case where certain mutations caused Wolfram syndrome without hearing loss. In this review, we asked the question of which WFS1 variants cause deafness. The functions of the wolframin protein include the regulation of cellular calcium via ER storage and negative stress response in the ER. Wolfram syndrome resulting from mutations in WFS1 classically causes hearing loss in addition to optic atrophy and different forms of diabetes. Current literature documents certain variants of the WFS1 gene that don't cause hearing loss, such as 2207G in exon 8 and 376G in exon 4. These both have all of the symptoms of classic Wolfram syndrome without hearing loss. The mechanism by which is partial functionality of Wolfram resulted in a less severe form of WS with no hearing loss. And on the bottom, we've got a figure that shows our confidence levels of what wolframin probably looks like. To better understand why some WFS1 mutant variants are responsible specifically for hearing loss, we also looked at studies showing variants that only cause hearing loss without the other symptoms of Wolframin syndrome. Certain examples of this are 2032T, 2033G, and 2022G. Both fully syndromic Wolframin syndrome and non-syndromic hearing loss result from WFS1 missense variants. The locations of the variants in regards to the Wolframin protein are significant. The variants causing Wolframin syndrome were distributed in all regions of the protein, while those causing non-syndromic hearing loss were clustered in the ER lumen domain, leading investigators to conclude that the variants causing hearing loss do so by a mechanism that involves too much ER stress. Most notably, non-syndromic hearing loss variants led to extra hydrogen bonds, which increase the wolframin protein stability, which suggests another potential mechanism to be studied. Furthermore, researchers also found that a glycine to arginine change at the same amino acid position resulted in classic syndromic wolframin syndrome. Our conclusions are that while examining different mutation variants of the WFS1 gene can give insights to correlated mechanisms resulting in sensory neural hearing loss, the precise mechanisms by which wolframin causes this disorder remain unknown. The locations of these variants can cause hearing loss are endoplasmic reticulum related, implying that ER stress plays a role in the development of hearing loss. Wolframin protein stability is associated with hearing loss. However, the molecular mechanism of this relationship is unclear. Amino acid substitutions at the same site lead to different disorders. Further studies are needed to examine the correlations between hearing loss and functional aspects of wolframin protein with respect to variant domains, molecular stability, and amino acid substitutions. I'd like to thank Dr. Pia Ghosh for her help with this assignment.